Hello, this is Nick, and this is the short 20-minute wrap-up on gene protein, production of proteins for your Biology 230 class. Basically what we're going to be looking at is gene expression, and we're going to be looking at basically how DNA codes for basically protein synthesis, and this includes two stages: transcription and translation. Now what we're going to show here is that we have a mutant deer here, and that is because that mutant deer either fails to produce a certain protein or produces an altered protein, and we're going to explain how that can happen. Again, there's a number of people who did work, and these are stories, backstories that you're going to need to know. Archibald Gerard is one I talked about in class, that he suggested that genes would dictate the phenotypes. And so this is looking at the basically expression pathways effect on genes and the protection of those pathways. Another group, Beadle and Tatum, exposed bread mold to X-rays, and basically what they found is this, and I'll get this experiment up there, but basically they had these classes of bread molds. They exposed them to X-rays to make mutations, and then what they did is that there is a pathway, basically a precursor gene, to go to ornithine, to citrulline, to arginine, and if you supplied just arginine, all of these mutants would be able to grow. But then they found that some of them were lacking a gene that would convert a precursor into ornithine. Okay, and these are one type of mutations. There was one gene that would prevent ornithine to being put into citrulline. And so if you look at it, this is a linear pathway that we have here, and they were able to identify mutations that would affect this. And so those are the genes. This kind of led to an idea of one gene, one enzyme, which is a very strong way to think about this, but not necessarily. And we'll talk about different types of genes a little bit later. You do need to know some terms. So transcription is the synthesis of RNA under the direction of DNA. Transcription produces what we call a messenger RNA, an mRNA. Okay. Translation is the synthesis of polypeptide, which occurs under the direction of mRNA by the ribosomes. And again, these ribosomes are the sites of translation. And we went over this in class, and you guys seem like you were pretty strong at being able to do this. In prokaryotes, mRNA is processed by transcription, is immediately translated, basically without any more processing. In eukaryotes, it's a little different in that the messenger RNA is processed. Okay, and we will talk a little bit about that processing. Again, we talked a little bit about the central dogma, which is DNA holds genetic information. That information converted into RNA, kind of a transient form, which is then converted into a protein. The idea in the beginning was that a gene code for a particular protein. This is looking at the transcription and translation in a prokaryote, which occurs in there. In the eukaryote, it's a little different in that the material needs to leave the nucleus. By the time a messenger RNA leaves the nucleus, it has been modified. It has a cap on it, it has a tail on it, and basically that's done for safety regions, so that the cell needs to know that that is a particular transcript that it can use, and that it's not a virus or something else like that. You know, people have been hacking information systems. Well, not people, but organisms. Biology has been hacking information systems for an incredibly long time. Okay, so it's one of the reasons why we have that, and we did talk about this. Okay. You do need to know a bit about the genetic code, basically how to read the genetic code, and we talked about this in class. We have about 20 amino acids, and they are coded by the triplicate code, and these codons. And codons, along with mRNA, are read by translation machinery in the 5' to 3' direction. Each codon specifies the addition of 120 amino acids. So actually this is made. It's not necessarily read So the mRNA is read in the 5' to 3' direction. Yes, and so that's what. So remember, we make things in the 5' to 3' direction, so we read them the opposite. And so here we have a DNA template strand. It is read in the 3' to 5' direction. This is the coding strand. This is what gives us the sequence of messenger RNA. Remember, this is RNA, and then this is translated. Okay, and so what we can have here is this is typically what we call an operon, where you will have a DNA molecule that has an operator region here, and we would make gene number 1, gene number 2, and gene number 3, and so on and so on. Codons are, we'll talk about what codons are in a second, but you guys need to be able to read this. So here we have the first site, second site, and then the third site is located in here. And so if we read a U, a U, a U, that's phenylalanine. That's what I was talking about. An A, U, a G, that's the start site. We talked about that. An A, U, a U is isoleucine. And the UA is stop. An UAG is stop. An UGA is stop. And so these, the caveman symbols here that I'm talking about are basically don't code for an amino acid at all. And so what we see is when we see a triplicate codon of CUU, that transfer RNA is going to have a proline on it. And that's very critical that you can be able to read this sequence and convert a messenger RNA sequence into the amino acid sequence. Okay, this sequence is universal, so we can take a firefly gene that codes for luciferase and express it in plants and get plants that grow in the dark. We can put the gene for the green fluorescent protein in pigs and again see that expressed in pigs. And so that is universal, and we were able to take the gene for insulin and put it in E. coli, and it works. And so RNA polymerase pries apart the strand and makes it. It starts at a region that we call a promoter, and it ends in a terminator, and we talked about that. The stretch of DNA that is transcribed is what we call a transcription unit, and this is essentially what we're going to talk about here. And again, we need to look at initiation, elongation, and then termination for each of these. And in your worksheet, you were asked to do that as well, and we will go over that. The promoter signal initiation of RNA synthesis. We have transcription factors which mediate the binding of RNA polymerase at the initiation of transcription. The completed assembly of transcription factors in RNA polymerase II bound to the promoter is called a transcription initiation complex. The promoter is called a TATA box, and so it's critical that you know what a TATA box is. And so what we would see here is that RNA polymerase binds on this. This is the actual start point, which is a bit up above from the AUG start site, okay? And there's some information that's required here that we know of, but it's going from the 5' to 3' direction. During RNA processing, both ends of the primary transcript are altered. So this is going to talk about. So we made our messenger RNA. Now what are we going to do with it? First of all, we see a 5' cap being put on it, and on the 3' end we put a poly A tail. These share several functions, and they facilitate the export of mRNA from the nucleus into the cytoplasm, and they protect the RNA from hydrolytic enzymes. That is, that anytime we have RNA just out in the cell will say, well, this is not from here if it's not from the nucleus, and we're going to break it down, and they help ribosomes attach to the 5' end there. And so typically what we would have, that 5' cap, that 5' upstream translated region, which is where the ribosome is going to bind. We have our start codon, stop codon, and then we have a polyadenylation signal and a poly A tail and the stop signal. we need to talk a little bit about these things that we call introns and exons and RNA splicing, and non-coding regions that are called intervening sequences. Not all of the sequences code for proteins. The exons are the actual sequences that are code for proteins, and mRNA splicing removes the introns and joins the exons, creating an mRNA molecule with continuous coding sequence. You say, well, why do we want to do this? And I'll talk about there's several regions, but basically it's a way for us to kind of modularize things and kind of move things around. But basically we take all of these exons and we put them together and we get a coding sequence. Okay. Spliceosomes are responsible for doing this, and you can have an idea of what a spliceosome is. Ribosomes are catalytic RNA molecules that function as enzymes and can splice the RNA. The discovery of ribosomes rendered obsolete the belief that biological catalysts were protein. And so whenever I told you that all enzymes were catalysts, but not were necessarily proteins, but not all of those are processes. Three properties of RNA enable it to function as an enzyme. It can form a three-dimensional structure. Some bases contain functional groups. The RNA is hydrogen bond with other nucleic acid molecules. The reason why we have this is that we have alternate splicing. And so if we look at a protein, we can actually see domains of functionality in protein. And so let's take the amylase that you used in class. It's going to have a carbohydrate binding domain. It's going to have a domain that actually functions to break the glycosidic bond. And so what we can have here is that these exons can code for certain domains. And then if you think about it, evolution can mix and match various exons in order to make a new and functional protein. Okay. If you take a look at Mastering Biology, they have a really good way of looking at this components of translation. I'm just going to go through it really quickly. Here is a ribosome, here's a codon, and then basically what's happening is you have a tRNA that's been charged by a tRNA synthase. Binding down here, we then see movement of this polypeptide, and so on and so on. And we'll have a video of that to look at. This is a tRNA. The tRNA is roughly L-shaped. It is made by a tRNA aminoacyl synthase, which looks something a lot like this. It requires ATP to be charged. But basically that tRNA is charged with a specific amino acid. The tRNA synthase gets charged first, then the tRNA moves here and we move it. And again, that's something that has to go relatively quickly. There's two ribosomal subunits. What we see, the large subunit and the small subunit. The small subunit binds first, and then the large subunit. You need to know what these parts are, and that's very, very critical to go over that. And on the large part, we have the E site, which is the exit site, the P site, the peptidyl tRNA binding site, and the A site, the aminoacyl tRNA binding site. And so what we hear is when we see the amino acid in the E site, it should be empty, and it pulls off. The growing site is in the P site, and next to that should be a charged amino acid, and we see that moves over, and there's this hydrolysis of GDP is required for that. So here we see in the P site a charged amino acid, and then basically what happens is a charged amino acid goes into the A site. We then see the hydrolysis of GTP, which basically moves the polypeptide over to the new site. All of this at the hydrolysis of GTP occurs, and that the empty site is now able to leave the E site, and it slides forward, and the process starts all over again. Again, a releasing factor would be the stop codon, which basically has no amino acid charge to it, and so what happens to peptide when it doesn't have anything to bind to, it's basically let go. In prokaryotes, once a messenger RNA is made, it will actually induce the formation of the ribosomes, and we will get production of a polypeptide kind of right away. This does not happen in eukaryotes. Proteins can fold and give us slightly different structures, and what we have is a signal recognition particle that binds to the signal peptides. The signal recognition particle brings the signal peptide and its ribosomes to the endoplasmic reticulum, in which case the protein, as it's being made, is being put right into the lumen. And so, again, this shows you how a secreted protein can be made. We're going to talk about mutations and point mutations right now. And basically what we see here is, here's the wild type gene. We can get a mutant type gene by basically altering one of the codons. And basically what happens here is that this single point mutation causes us to go from normally putting a glutamine here to putting in a valine. And this is what causes sickle cell. And there's a number of different types of mutations that we can have. Again, a silent mutation is basically when we mutate the third codon, in which case we're going from a glycine to a glycine. That's really, even though it is a mutation, we don't see it's a silent mutation. A missense mutation is where we put in another amino acid. A nonsense mutation is where we put in a stop. Now, frame shifts can also cause mutation. So if we put in or take out anything less than a series of 3 amino acids, we're going to mess up the whole sequence kind of down the line, and that is critical. So we talked a little bit about this, and that's basically about it for today's lecture. Thank you.